Good morning, everyone, and thank you for joining us. I'm pleased to be hosting today's conversation with Matt Ebmeier. Matt and I have worked together for over 2 decades in a lot of different iterations, and now with each of us having our own companies, which is pretty exciting. I say that I am the AD to his FD, the analytical development to his formulation development. So Matt's the founder of ProTopical Solutions, where he develops and manufactures high-quality topical products with a clear path from concept through commercialization. Before we dive in, a quick housekeeping note. This webinar is being recorded, as she just told us, and we encourage you to submit questions through this session using the Q&A feature. Matt, to get started, can you tell everyone a little bit about yourself and a little bit about ProTopical Solutions?
Sure. Good morning. My name is Matthew Ebmeier. I am, as Wendi had mentioned, the founder and co-owner of ProTopical Solutions in Loveland, Colorado. We are a development and manufacturing site for topical and semi-solid products. We serve the pharmaceutical, OTC, and cosmetic industries. A little bit about my background. I have a little over 20 years of pharmaceutical R&D experience. I have 17 FDA-approved drugs for prescription that I took personally from the benchtop all the way through FDA approval and commercial launch. And then as director of formulation development, I had the entire portfolio of about 38 approved products. And that was for both topicals and some oral products as well. I actually started my career as an analytical chemist. So this topic is near and dear to my heart, and I'm looking forward to today's discussion.
Excellent. Well, to kick it off, the first question I have is, can you explain what is formulation development?
Sure. Formulation development is basically taking a concept or an idea. Typically, if you have an API or an active pharmaceutical ingredient and a target indication in mind. So a lot of what we'll do is we will evaluate that drug, evaluate the indication, and then build a product that is specifically designed for that indication and for that drug. From the drug standpoint, we want to make sure it's both chemically and physically stable. And then from an efficacy standpoint, we want to make sure that it demonstrates clinical efficacy.
Excellent. So what are some of the challenges over the years that you've seen? What are some kind of formulation issues that you've had or you've seen?
Well, there are many. You typically learn more from your failures than from the successes. We've seen issues where the drug is in extremely low concentration. We've seen issues where the drug is just not really soluble in a lot of compounds. Typically, if development goes relatively smooth, if it's either hydrophilic or hydrophobic, so it dissolves in water or dissolves in oil. It's kind of those in-between compounds that typically give us a little more development time in order to make sure that we have a product that's both stable and efficacious.
Yeah. I think we've shared in some I want to say nightmares of issues with either formulations or the analytical side of the formulations. Really low. One I remember was a really low ointment product, like 0.005% in an ointment base. So it was really challenging. Yeah,
it was. It was also challenging to manufacture and make and ensure that we had complete homogeneity from the bulk all the way through our finished products.
Yeah. So how is topical formulation different from, say, formulating a solid oral or a liquid oral or an injectable?
That's a great question. So I think the development pathway is typically relatively similar all the way through. You really want to understand your API or your drug. You want to understand what it's compatible with all the other excipients that you want to use in your finished dosage form. So whether it's a tablet or a topical, that's really phase one of development, is understanding your molecule and how it presents into a finished product. And so there are a few unique, characteristics that come down to semi-solids or a cream or a lotion or anything along those lines, mostly because you have a thermodynamically unstable system. Water and oil don't mix. And so what we're trying to do is ensure long-term stability by using different surfactant systems and emulsion characteristics and microstructure in order to achieve long-term chemical and physical stability. So they are somewhat complex systems.
Yeah. So it
Compared
kind of
to,
comes down
say,
to, yeah, sorry, go ahead.
yeah, compared to, say, a tablet where you're going to blend your powders together and then compress. So that's,
I think they all have their own challenges. I think the semi-solid, the consistency, it adds kind of it's like a user aspect to topicals that you don't see when you're looking at solid orals and things like that. Yeah.
yes.
So, you know, we talk about quality by design a lot, and I'd be curious to know kind of where do you bring that in? Where do you start with like the quality target product profile, the QTTP, and all of that? When does that kind of come into your development process?
And to me, that's very much one of the very first steps. And that's really understanding what kind of product you want to create. It really kind of creates the framework from the start to the end. And in that QTPP, you're going to lay out what your indication is. Is it acne, psoriasis, rosacea, or, you know, is it like for athlete's foot if you're more on the OTC level, or if you're targeting anti-aging or wrinkling on the cosmetic side? Really understanding your indication, the skin type. Is it compromised skin or is it intact skin? And kind of the potential irritation levels that can come about. So you're really laying that out in that original quality target product profile, the FDA and their acronyms.
Right.
So it really helps create the framework for the development start to finish.
Okay. Okay. So, you know, the QTTP feeds into the CQAs, the critical quality attributes. How do you, like, what are some of the more important CQAs that you see? Like, what do we see for topical CQA that we don't see elsewhere?
So a couple of the ones that come to mind are going to be your physical stability. So is it maintaining its, is it still a off-white opaque cream, you know, throughout the shelf life and stability of it? Additional steps would be things like pH, because typical creams and lotions contain a continuous phase of water. And so that can affect your physical stability. So if you're getting shifts in pH over time, that can affect your chemical stability. Viscosity is another issue that you would only typically see in a semi-solid. Typically, if you've got a robust, stable emulsion, your viscosity won't shift or change over the course of time. That's really a key attribute, because if you tend to lose viscosity, you can actually see things like phase separation. And when you start to see phase separation, then your drug has a tendency to migrate into different areas. And that can affect the homogeneity. So if you think of, you know, like your tube of cream or ointment, if you ever take the cap off and you squeeze something out and you get a layer of water that's coming with it, well, if you've got a water-soluble drug, you've got a super potent area that you just dosed yourself with. Or if it's more oil-soluble, you might be essentially getting the placebo version versus the active, which is now being concentrated in the oil phase.
So making sure that you don't have phase separation in a topical, that's the biggest, I don't want to say challenge, but it's the most important thing, is making sure that it's not going to separate so that you get consistent dosing. Yeah. I always tell people phase separation is like your grandma's lotion that she had in her closet for 10 years, and you go in and you get some and it all just comes out water and it's kind of gross. Yeah. So to kind of feed on that, you mentioned like creams and lotions and stuff like that. Can you walk through what are the differences between like a gel, a cream, an ointment, a lotion, like specifically like a cream and a lotion? They're kind of similar.
Yes. So we'll start with cream and lotion because that's pretty similar. A lot of the product development that I've done, the biggest difference between a cream and a lotion is really the amount of residual oils that are in that product. So if you figure a lotion product is roughly about 15% oil phase, both liquid and solid oils, and then some surfactants. So roughly 85% of the product that you're applying to the skin is actually water. And so as you put a lotion on, it's going to take a few moments for the water to evaporate off. And then you're going to leave about that 15% of oil phase that's actually going to give, the lotion feel. That's going to help you, create a boundary on the skin to prevent epithelial water loss. So a cream is very, very similar. We're just increasing the amount of oil. So if you figure a lotion is about 15% oil, cream could be somewhere between 30 and 50% oil. And it's just how much residual product are you actually leaving on the skin?
They're both emulsions, right?
Correct.
And
They're
what
both oil and water.
oil and water emulsions? Is there a product type or an API that you would formulate into a lotion more than you would into a cream? Is it like a solubility thing, or is it a feel thing? Or?
It's a feel thing, and typically based on the indication. So like if you've got a condition where it's very widespread on the body and you really want to be able to cover a large surface area, versus something, say, like a psoriatic plaque or even rosacea, something along those lines where you're actually covering smaller surface areas, then a little heavier product actually works pretty well. I typically go more towards lotions when I need to cover surface area.
Okay. So then the other two were I think gels and ointments.
Yes.
Can you talk a little bit about those two?
Yeah. So gels are typically when you've got a API that loves water, is very water soluble. It works great for those. Gels can be very light, and for things like acne, they work great because for acne, you don't want to actually introduce additional oils to your skin. So you want something that actually kind of strips the oils away. So for indications such as acne, I typically target gels. They work very well.
We wouldn't
for
want
that
it to be
indication.
ointments for acne.
Right. You would never want to do an ointment for an acne product.
Okay. And then ointment, I think, having it be that oily, typically we would see that for things like psoriasis, right? Where you want it like
psoriasis, eczema, conditions like that where you've got dry, cracked skin. You really want to be able to hydrate the skin as you're also treating it with your chemical compound.
Yeah. So is there any of those different dosage forms that are more challenging than others to formulate or manufacture? I know it's kind of a broad question because everything has its own challenges, but
yeah, everything always has its own challenges. I would say emulsion science is typically your hardest. Everything else, you're essentially creating a true solution. Say a gel is essentially a true solution. Does everything dissolve? Does everything stay in solution? And then you just need to typically use a polymer in order to thicken the gel so it squeezes out of a tube. Ointments are kind of similar, where typically a base is petrolatum USP. Most people know it as Vaseline. And so you're typically incorporating your drug, which might even be emulsified within that ointment base, but it's a much more stable system. And you don't typically see phase separation. I mean, we have in past products, it does happen. And then it just needs to be resolved.
Yeah.
But creams and lotions are typically more complex.
So we're kind of walking through the development process. So we've talked about like how do you pick the dosage form, like cream, ointment, gel, that kind of stuff, right? So once you move from that, I've got my QTPP, right? I've got an idea. I know what the indication is. I have an idea of what the formulation's going to be. what are the first formulation studies or experiments that you do to when you're getting started?
So the first steps are going to be relatively the same for any R&D program. You really want to understand your drug and your indication. Those are the two things that I focus the most on. One is considered more on the paperwork side, where I might be evaluating scientific literature, I might be looking at patents, other products are on the market for such an indication that might be competition in the future. So that's kind of the paperwork side. And then once we start on the lab bench and we start doing the lab work, it's really understanding your molecule. What is it soluble in? What is it stable in? So I'll make binary and tertiary systems. So if I want to dissolve it in a compound such like propylene glycol, I'll take the drug, I'll dissolve it in propylene glycol. All of these are done on the bench. And then I would go to Wendi for analytical development, and we would test those samples for T0. Are we getting the amount of drug back that I know I put in when I compounded them? And then we'll go ahead and put those on accelerated stability, typically 40 or 50 degrees C, evaluate them for at least 2 weeks time. And what we're looking for is chemical degradation. So is our API compatible with those specific excipients? And that can tell you a lot from the standpoint of, will you have a stable product in the future? And so you're just kind of, think of it as laying the ground blocks of your product development. So once I have, I'm sorry, go
real
ahead.
quick on the excipient compatibility. If you have a failure in excipient compatibility, let's say that you're looking at a preservative and the preservative's impacting the API, is that like a deal breaker for that excipient, or what do you do
Typically,
next?
we would shift out the preservative system if we're seeing compatibility issues. I see it often with solvents. We call them penetration enhancers in the industry, because that's what you're trying to use to get the drug through the different layers of the skin. And a lot of times we will see stability issues. You can even see color changes. I've seen things turn pink at 40 degrees C.
Yeah.
So it's sometimes they're obvious and sometimes they're not. But without that analytical component to tell you you're compatible, you might be using an excipient that could be having dramatic chemical stability issues on your product.
Mm-hmm. So once you've kind of looked at the excipient compatibility, how do you develop like the prototype, the ratios of all of the excipients? I used to joke that we had a formulator and he would be like, and I'm dating myself, but he'd be like Johnny Carson, and he put like an envelope to his head, and he would say it's 15%, like polysorbate. I'm making stuff up, but he would do that. It was always a joke, and it was like somehow you all just know what the ratios need to be. How do you figure that out?
I had a really good mentor for about 6 years. He was one of the top formulators in the nation. Pretty much every patent that I pulled had his name on it. And so I really had that mentor-pupil sort of situation for about 6 years. And he kind of taught me general use levels. Now, there's other guidelines that are out there. For example, the IID listing on the FDA website. That'll tell you for each and every specific ingredient what the maximum concentration you can use from a percent weight, because that has been tested clinically. And so that's always another good rule of thumb. So you kind of start pulling in all these different pieces of information. Some of it might be, knowledge that you've gained from over time. And then you kind of really start narrowing down your ratios and your concentrations. And I'm a big believer in only using excipients that need to be within the product, rather than trying to add a whole bunch of additional ingredients that just makes your ingredient label huge. I see that a lot on the cosmetic side. I tend to take a little more clean approach and then add things in as, say, a client might be interested to to have a few extra ingredients. But
maybe some IP protection of some kind, or yeah.
And that's a whole nother discussion. So, and IP protection is always great. And your best bet is originally around your molecule. That's your safest form of IP. And then after that, there's several tricks of the trade in order to patent your formulation so that no one else can touch it. And if you have a product that falls into that category, it's one of my favorite things to do.
Yeah, try to figure that out. What is the best patent protection for a topical? Like, what is the best thing that, what is the thing you can get the most IP on?
First and foremost, it's the molecule, if you have a unique molecule. After that, then you can move into formulation technologies where you're actually creating something new and something novel. And then you're actually going to patent that formulation platform that you could then use for multiple other compounds. That's probably your second strongest. And then your third would be what we call a picture claim patent, where here's our formulation, here's all of our ingredients at this concentration with this active compound. You can't copy it. So those are reasonable patents.
So I think one of those would be like, if you came up with a novel like penetration enhancer, a novel system to help deliver maybe to a specific, and I'm not a formulator or a clinician, but a specific part of the skin, right? I'll let you kind of talk about that. But
yeah, so if you develop something that, say, targets the epidermis or the dermis layers of the skin, and you've got a novel delivery system, that's definitely patentable.
Yeah. So back to we talked a little bit about preservatives. What are some preservative systems that you use in topical? Like, when do you need a preservative?
Anytime you have water present, you definitely need a preservative. So, for example, ointments are self-preserving. They're an anhydrous system, and microbes can only grow if you have water present. So the preservative side is the bar is much lower when it comes to preserving, say, an ointment versus a lotion where you've got a large quantity of water. You're going to want to evaluate your pH. You're going to want to evaluate potentially multiple preservative systems. There's some natural ones out there now that I've been using on the cosmetic side. There's very standard ones that we've used on the pharmaceutical side. And then parabens. Once you parabens are a big one. Personally, I think they work great at really low concentration. But a lot of people are a little afraid of parabens. So we've kind of introduced some new preservative systems for the products.
Yeah.
And then in order to evaluate that, you're actually going to do what's called preservative effectiveness testing, PET testing. And what that's going to do is that's going to challenge the finished product itself. So they're going to take, let's say, hypothetically, we're making a lotion. They're going to take that lotion and they're going to plate it out. And they're going to reduce that concentration of your product all the way down until it actually starts to grow bugs.
And they're plating it for micro, right?
For micro, correct.
For micro, yeah. Just like general, total aerobic. Do you think? I think, yeah. Okay.
Yeah, anaerobic and aerobic. And you're just looking to see what the fail point is. And is your system perfectly preserved? The other test that we, evaluate is MLT testing, so microbial limits testing. And what that's looking at, that's something that you're only going to do at, T0, essentially after you've made your product. And you're just looking to make sure that you didn't create a contaminated product either through the excipients you used or the equipment that it was compounded on, or water. Water is always the tricky one. So,
that made it, right?
Yes.
Yeah. It didn't get contaminated. And microbial limits, I think that's like E. coli or that kind of stuff. You're looking at specific microorganisms.
think there's 5 or 6 specific microorganisms that TIS evaluates for.
That I think are concerning for topicals, right?
And
Yeah.
patient safety.
Yeah. So there's something else that gets added to topicals a lot, and that's antioxidants. When is it necessary to add like a tocopherol acetate or something?
A lot of times I'll add tocopherol acetate to almost every formulation, just because having a little antioxidant in it, and vitamin E works great on the skin as well. So you kind of get the best of both worlds. I've worked on some pharmaceutical compositions where they have a very strong oxidative pathway. And so by adding in an antioxidant, you're actually helping to, prevent that pathway and extend your chemical stability. So it's a lot of times I won't go to a strong antioxidant, for example, BHT, BHA, unless I need it. And if it's discovered during the early development that we've got a chemical stability issue due to oxidative degradation, then we'll go ahead and add in those powerful antioxidants that work very, very well at a low concentration.
Okay. And then, this analytically gets challenging when you've got preservatives that you're testing for. Or vitamin E is a challenge. Yeah. Yeah. Not super straightforward. So once you you've figured out what excipients you want to put in there, you've figured out the ratios, you've got some prototypes, then let's talk a little bit about in vitro testing. Tell us a little bit about, talk about Franz cells. Tell us like what you do there, what you're looking for, what kind of data you get.
Okay, so after we've got our initial prototypes, so we might even have prototypes that are a cream, a lotion, a gel, or an ointment. Typically not will be a little more focused based on the indication we're looking for. One of the things that I'm going to do in those multiple prototypes is I'm going to evaluate different penetration enhancers and different ratios of those penetration enhancers. And the only way to really screen formulations for the one that you want to move forward clinically is to evaluate them. Well, one of the most cost-effective ways to evaluate them is what's called IVPT testing, so in vitro permeation testing. And it's done on a Franz cell. I just happen to have an old prototype here that looks very similar to this, where you actually have a receptor solution in one side, your product goes in on the top, and then you can either have different types of barriers that you're trying to work your way through, either from membranes to human cadaver skin. And what the system really does well is it allows you to compare your formulations against them or against each other in a very, very controlled system. So, for example, an ointment might penetrate twice as well into the epidermis and the dermis than, say, a gel, which just dries out and sits on the very top of the stratum corneum. So it allows you to really put a head-to-head comparison against your different prototypes. And once you get that data and you have a chance to evaluate it, then you can kind of pick and choose for your next round of optimization, looking at your skin permeation data, your penetration enhancers, and your target indication. And it really helps you narrow down to get to essentially a final formulation.
I think that different diseases affect different parts of the skin, right? And so are you able to target with your formulation really where it delivers?
It depends a lot on the molecule. Every molecule can create different challenges, based on its molecular weight, based on its charge, based on its, hydrophilic or hydrophobic solubility. Some molecules are much easier to target into the deeper layers of the skin, and others are more difficult. And it's the ones that are more difficult that we find to be the bigger challenge, and just takes a little more time from a research and development standpoint to really narrow down so that you get the best product for the best indication.
how do you figure out how deep it penetrates?
So
And
so for example, for IVPT testing, when I outsource this, I used to do it once upon a time when Wendi and I worked together.
yeah.
And I actually did that in the lab, but now we outsource that. And so the report that I'll get back will actually give me concentrations of the API in the stratum corneum, in the epidermis, and the dermis, and then also into the receptor solution. So how well is that drug permeating and working its way through the deeper skin layers? And they're actually fractionating off the different skin layers and sending them for LCMS analysis, because a lot of times we're looking at relatively low concentrations. But it allows you to really see how your dosage form helped the drug permeate through the skin.
Do you ever develop a topical that's intended to be delivered systemically? Have you developed one of those?
Yes. We didn't end up pushing that product forward, mostly because it was a hormone and it had presented its own difficulties from a manufacturing standpoint. But for example, testosterone, estrogen, those are both hormones that are designed to penetrate through the skin for systemic delivery. And I can honestly say that testosterone, I have never seen a molecule penetrate the skin as well as testosterone.
Really?
Relatively simple formulation, and it was amazing how well it actually permeated from a systemic standpoint.
So like on the other end, have you ever had one where it just sat on the top of the skin? You couldn't get it into the skin.
It's tricky. Yeah.
You throw like a whole bunch of penetration enhancers at it and just.
Yeah, we'll throw a lot of times, you always start out with a very aesthetically pleasing product. And then when you start hitting some of those roadblocks, you might end up, taking the gloves off and really getting into some penetration enhancement and really starting to try and compromise the stratum corneum in order to get the drug to penetrate through the skin.
Yeah. You kind of have to break up the connections a little bit in the skin to try to get it to. So, I had a question about there's some topicals that we've developed that are suspended, and like scientifically having a suspension, so that means like the drug is solid, it's like particulate, right, in the topical formulation, but yet it's still efficacious, right? So how does that work? Is there a certain amount that dissolves? Does it dissolve when it hits the skin?
A lot of times what happens when you have a suspended product, and let's stick with the cream model, where if you have a suspended drug, I saw that a lot in corticosteroids, typically what you'd get for a rash or something along those lines. The way that you maintain chemical stability is by suspending the micronized drug in the emulsion. Then it's kind of the concept of the product in the tube versus the product on the skin. And so the product in the tube, you've got suspended crystalline drug to maintain your stability. Then when you apply the product to the skin, you are flashing off all the water. So let's say 70% of the product is water. That water flashes off. Now you've got that other 30% that are residual solvents. And typically you're picking residual solvents that dissolve that API when it's on the skin. So something like propylene glycol, which is a great penetration enhancer, and dissolves a lot of different products. So that residual layer that's left on the skin itself is actually dissolving that crystalline drug and allowing it to penetrate into the layers of the skin.
Okay. That's kind of interesting. Yeah, I always kind of wondered that, if it's a solid, you're certainly not grinding it into your skin, right?
No. And, crystalline API is typically not going to work all that well as far as like a skin condition. You really want to get things into solution.
Yeah.
Whether it's in the product or on the skin.
So once you get that all figured out, like you've done your IVPT, which is the in vitro penetration testing, so
Permeation. But
permeation
yeah,
testing. To get that done, then it's kind of maybe time for the develop the process, like how you're mixing it together. And I think, when ointments, are pretty straightforward. You're dissolving up, heating everything up, and mixing it together. Hopefully it's pretty straightforward. But emulsions are a lot more challenging. Can you talk about how you develop the process for making an emulsion, and particularly how do you make it so that it stays together and you don't see that phase separation?
yeah, absolutely. So a lot of that's going to start on the benchtop itself.
Mhm.
So when I'm developing a product and a process, I'm already thinking forward to how we're going to manufacture it from a commercial scale. What equipment do we have that we're going to use? How will, what I'm doing on the bench correlate to something that's going to produce, 5,000 tubes or 20,000 tubes? So you're really trying to keep those same concepts as you're working through the process. So I don't want to use a technique on the benchtop that I know I can't use from a commercial standpoint. And once we kind of get through the benchtop formulation work, we're down to essentially a master formula. Now we need to start looking to scale up for, say, clinical supplies. Well, the next step is we start doing what we call engineering studies. The first one we're probably going to do is going to be with a vehicle, so without the active ingredient. A lot of that comes down to cost and complexity, and it really kind of allows you to see how the product behaves on the equipment that you're using. So as you can see behind me, we've got manufacturing equipment where we can actually scale these products up. And so we're going to create that engineering study where we might be making anywhere between 50 to 200 kilos of cream. And we're going to, then once we've made that product, we're going to evaluate it from a physical and chemical standpoint. And then once you kind of get your process all nailed down, then you're going to want to go back and make it with your active compound that's going to create your finished product or your phase 1, phase 2 clinical supplies.
So how do you develop because I know from all of our years working together that you can put a lot more energy into something on the bench, right, than you can, so how do you go from a bench scale to that pilot, clinical lot size, right? How do you figure out what how do you correlate the two, I guess, to make sure that the emulsion's stable?
So there's a couple different ways that you can go about it. You're absolutely correct, where I can put a lot more energy in on the bench than I can at full scale.
Like it's
You
like
can,
a blender that you would use,
Mhm,
right? Yeah, you
right.
really blend it.
And we can do the calculation. We can calculate what the tip speeds of the mixers are and try and correlate that from small scale to pilot scale all the way up to commercial scale.
Mm.
Uh, as a formulator, I've always felt that, when you make these products on the bench and say I make it 20 or 30 times, you get a really good feel of what looks good and what doesn't. And then being there to witness the product actually take shape as you move it into pilot scale, into commercial scale, you can tell a lot.
Yeah.
And then, of course, we're going to do analytical techniques as well. Wendi can test physical and chemical stability. We can look at things like droplet size. We can look at things like, the microstructure essentially,
Mm.
and see how that correlates as we scaled from benchtop through pilot all the way up to commercial scale. And
So
that kind of helps build that confidence as you go.
yeah, so you can almost visually see with droplet size whether you're getting a consistent emulsion.
Yeah,
Look at the
microscopically. Yeah.
yeah.
We'll use a
Okay.
microscope and
Yeah. And then, we've been talking a bit about emulsions, maybe not as much as you probably talk about emulsion, you know what I mean? Like emulsions are a big part of topical product development, right?
they are. They are.
Yeah. And the, we talked about phase separation and all of that kind of stuff. What is it in the emulsion that drives the stability of the like the microstructure stability that keeps it from phase separating over 2 or 4 years on the shelf?
Sure. So there's a couple key components, and in my opinion, the ones that matter the most are the surfactant system that you're using. So water and oil don't mix. We've all seen that before. So really what you're using is you're using a molecule. It's called an amphipathic molecule. One side is hydrophilic, loves water. One side is hydrophobic, loves oil. And it's all attached in the same molecule. So when you create an emulsion, you're essentially creating small oil droplets within the continuous phase of water. And the entire surface of that oil droplet will be covered by your surfactant system. I always like to use 2 different surfactants rather than one. It seems like you get a better interlocking of those surfactants. It gives you a better long-term stability. Now, I can have a perfect surfactant system, and when I go to make it on the bench, if I just use, say, like a paddle mixer, or even if I just try and blend it by hand, let's say I took a whisk and just tried to whisk it up, I am going to create those oil droplets within the continuous phase, but typically they're irregularly shaped. They can be kind of a globular, unstable looking underneath the microscope. And those are almost always the first ones to phase separate. And it's just because you didn't create that good, stable emulsion system with really consistent little oil droplets. So what I'll work on the bench is using equipment that's specifically designed for doing that. And what I'll do is we'll use a homogenizer, is a great mixer. And what that does is it gives you very, very uniform oil droplets. And the smaller and the more uniform they are, that's typically your greatest physical stability. And I've seen products that I developed, 15 years ago that I still have samples of that are perfectly stable emulsions to this day. So that's one of the key characteristics, is when you're actually looking into the microstructure of the product.
Yeah. So we talked a little bit about the excipient compatibility and looking at that, maybe storing that for a couple of weeks at temperature. Tell me about how you utilize R&D stability testing to kind of evaluate your prototypes.
It's, in my opinion, one of the most key pieces of development. And literally R&D stability testing is telling us whether or not we have a chemically stable system. So there's 2 things that are death to a formulator, in my opinion. And that's creating a product that's not physically or chemically stable. Whether or not you've got to start shortening the shelf life. We've seen that in our career, where we were creating a lot of generic products at the time, and the generic that we copied almost always has the exact same issues that we did. And we might start seeing chemical stability issues where we're falling out of that, say, 90 to 110 assay claim at 18 months instead of 24 months. And so we start reducing the shelf life, and nobody ever wants to do that. The other death to a formulator is essentially if we have a physical failure where we actually see phase separation, and you get the oil in the water goes back and starts layering itself out. Well, where is your drug? How does that happen? And then you lose API homogeneity within the finished product, and that's also a critical failure.
I think we saw there was a phase separation issue that we saw that was ointment in an ointment. What did you call that when it was a different term than phase separation, when the mineral oils coming out of the oil or out of the
syneresis.
Syneresis, yeah.
And
that could be a big problem if the drug's concentrating in that mineral oil, right?
That's correct. And so with one of those products, just having pharmaceutical grade petrolatum USP fancy Vaseline when it would come in a 55-gallon drum, we would open up those 55-gallon drums and we'd have a layer of mineral oil across the top. So petrolatum is essentially two components. It's mineral oil and it's microcrystalline wax. And so that combination of the two is really how you get that nice semi-solid oil. Well, some grades can actually bleed out a little bit of mineral oil. So if your initial component, if your initial raw material demonstrates a little phase separation, it's something that you really need to evaluate from the R&D phase to ensure that it doesn't happen in your finished products.
Mm-hmm. Yeah. Yeah, I thought that was interesting. And one thing that I talked about in previous webinars that we did was the criticality of assessing different batches of a raw material. So I talked about there was a situation where we had only one lot of petrolatum that we used for all of the development batches, and then we went to make the commercial batches, and all of a sudden our viscosity, we couldn't meet viscosity, right? And so, having that kind of rigor and looking at that as part of the development, I think is really critical.
I completely agree. And I'm a big believer in using USP NF material wherever possible, just because it ensures that the product that I make today is the same product that I make 5 years from now. And if you're buying your raw materials from some random place, and you can start to see a lot of variability in your finished product. And the investigations get really complex.
Really complex, and it's almost a showstopper sometimes until you can get a spec change through, right? Yeah. Right. What kind of considerations do you have when you're sourcing your raw materials?
Confidence in the supplier is always one of my key attributes. And then if it's USP NF material, I feel very confident. And as you start working your way down to materials that are not USP NF, they might be, if it's on the cosmetic side and I'm doing essential oils, I want to use the most reputable company I possibly can.
Yeah.
And will provide consistent raw materials to make the product.
And do you do robustness studies with them, to kind of evaluate what, do you try to get, do you call them and ask them for, can I get 3 lots of this raw material kind of thing? Or how do you do that?
From a quality by design standpoint, that was actually that's a checklist for an NDA, for the FDA. And so a lot of your more reputable companies that work with the pharmaceutical industry a lot will have those packages for you. So you call up, we're looking at this raw material, this grade. Do you have 3 different lots that you can send us for QBD evaluation? And a lot of them do.
Okay.
What I've seen on the cosmetic side is if you call up and ask the same question, chances are that it's a little more random. And it's rare that I've seen that QBD kind of mentality on the cosmetic side.
Do you find that it's easier to formulate a pharmaceutical than a cosmetic because of that? Is it easier to be consistent?
Definitely easier to be consistent.
The criticality of the consistency it's not there for a cosmetic like it is for a pharmaceutical. You might not have the specifications for viscosity or.
Right. And you might not have the specifications. It's the development timeline for, say, an NDA product or a pharmaceutical grade product is more like 8 to 9 years, where on a cosmetic product, it might be more, 6 months kind of time frame. And the one nice thing about a cosmetic is it's much easier to substitute a raw material supplier or even make changes to the formulation in and of itself. So you're more rigorous on the pharmaceutical side, but you have more flexibility on the cosmetic side.
I think we're seeing some GMP requirements coming out for cosmetics. Is that right? We didn't really have one before. Right.
So GDUFA went into place on January 1, 2024. And, I'm sorry, it's not GDUFA. That was Pharmacide. MOCRA is the cosmetic side.
It's like GDUFA.
Where,
yeah.
Yeah, sorry about that. So MOCRA actually is where the FDA came back and said that all products need to be manufactured under CMG or CGMP, which is good manufacturing practices. And really what it's doing is it's forcing people to really understand their products, understand their raw material suppliers, create specifications around each and every excipient, understand their product, their process, their packaging, their preservative systems, and really kind of taking a lot of the same concepts that we've worked with on the pharmaceutical side and sliding it down into the cosmetic realm. And so one of the advantages of me creating ProTopical Solutions was, having spent over 20 years making products for pharmaceutical companies, it's really easy to work backwards into the cosmetic realm versus, say, having never been exposed to GMP manufacturing and all the additional paperwork and that goes into it, and then bringing those systems up to the FDA standard for CGMP.
Yeah. And I think, like, how does then you've got cosmetics on one side, you've got pharma on the other side. Where does the OTC, because it's OTC drug, it's still drug. Where does that fall in that spectrum? Is it full pharma? Is it?
A lot of times they're treating it very much as full pharma. The biggest difference with OTCs is they fall under the FDA OTC monographs, where it's a certain drug at a certain concentration for a certain indication. That is really the three criteria. What they do give you a little flexibility around is the formulation that you put them in. So like I have a little more leeway on the excipients I get to use. But if we're claiming, for example, 1% adapalene, well, that 1% adapalene needs to be, it's a 0.1%, has to be in there at that known concentration in that dosage form to be used for acne.
Is the dosage form like it's an adapalene gel? It's
Correct.
not so it'll say very specifically the type of topical, or
Correct.
Like, but as a manufacturer, don't you still need to follow like drug GMPs for an OTC? It's because there's like in the solid oral space, there's the OTC, like more the the supplement, right? Dietary supplements under DSHEA, right? There's specific GMPs for that. Right. And then there's the drug. On the topical side is cosmetic, which isn't supposed to have, any. Yeah. So it's a little bit different.
It is. Like on the cosmetic side, you can't have drug claims. On the OTC side, which is one step up, you can have drug claims, but you have to follow the FDA or the monograph that's out there. And then on the pharmaceutical side, that's either, NDA pathway or ANDA pathway or 505B. There's several pathways.
And I guess there's like tons of OTC or solid orals too. So
aspirin, allergy medication. Those are all OTC products.
Yeah. So one of the things that I've been saying a lot in talking about analytical development, the first webinar was specifications. We did analytical development. I just did last time impurities, ICHQ 3A and B, right? And all of these, I'm really touting the need for analytical development to get involved early, to start the development early, right? And kind of the purpose of that is to make sure that the data you're generating in the excipient compatibility or in the R&D stability can be read through all the way to commercialization, right? Do you have any kind of thoughts on that? Do you agree? I guess, do you have an opinion on that?
Yes, I say I absolutely agree. I think it's critical. That way we have real-time information as we're developing these products and working them towards commercial. So I remember back in the day when we choose to go with a new API that we were potentially looking at, not only one of the first things I would do would pull a sample and walk it across the lab for Wendi to test so that you could start your method development at the same time that I'm starting the benchtop work. So that we're trying to hit that correlation. So when we're looking at excipient compatibility, we're on the same track. And the more evolved the methods, the more evolved the formulation.
Yeah, we had a situation once where we were trying to validate a method, and it was a different formulator. It was a short-lived formulator, but the product that Matt always gives me stuff that's right on 100% for my method validation. And I just kept getting 97%. And I skipped a holiday weekend trying to figure out what was wrong with my extraction, only to find out on Monday that, yeah, no, it really is just at 97%. It's very frustrating, but yeah. So, so back with that, I think that one of the things that we had, and this actually just came up with one of our customers here, is method validation really has to happen. It should happen on the final formulation. So we always, we would have we had a gate that we had that was, is this the final formulation? Are you sure it's the final formulation? Are you 100%? Because if you went in pharma, if you go in and you tweak one of your excipients and you go from 5% to 7% on something, then we would have to go in and revalidate our method. We'd have to do some, if not a full revalidation, we'd have to do some kind of bridging study. So it was always like, no, really, is this the final formulation? Is this the final formulation? And after the third time of me asking, you'd be like, yes. And I would feel convinced that we could go forward with method validation at that point. But we, you give us method validation or method development supplies. As soon as you have a prototype, we would start in on the method development and assessment to see what we saw. We'd throw some in the stability chamber, start looking at R&D stability and all of that, see what kind of impurities we can form and create, a little bit of force degradation and all of those kind of things. So, well, Matt, we've only got 2 minutes left. I have one final question for you. Matt, what is your formulation philosophy?
Make products that help people.
Products that help people. It's about the patients, isn't it?
It's always about the patients.
Yeah. I think a lot, it's the do no harm, right? We certainly don't want to do anything that's going to harm somebody, right?
And that's first and foremost. Yeah.
Yeah. Yeah. Do no harm and help people. Give them something that's going to work for them, that's consistent. So, well, Matt, I really want to thank you so much for coming on here. I appreciate your perspectives and sharing your experience with us. I think that I talk a lot about analytical development, but there's no need for analytical development if we don't have a formulation development. We've worked really closely over the years, and I think that a close connection between formulations and analytical is really what drives rapid product development. You can develop it a lot faster if you have good data, right? But then also quality products that are coming out of it. So if today's discussion resonated with you and you're considering outsourcing topical formulation or manufacturing, I strongly encourage you to connect with Matt directly at ProTopical Solutions. Matt, what's the best way to get a hold of you?
You can find us on our website at protopical.com.
Excellent. So thanks everyone for joining us. We appreciate your time and we look forward to continuing the conversation. The next webinar is going to be on April 1st, April Fools, and we're going to be talking stability, a deep dive into ICH Q1A. All right. Thanks, Matt.
Bye.
Bye.